Swayam Prabha. Digital India. Educated India.
Hello learners. Today we will continue with our discussion on the topic Products of Evolution. This is the 7th module from the paper Evolutionary Biology, and this is the second lecture under this topic. In the previous lecture we have talked about what do we mean by products of evolution, what are various microevolutionary and the macroevolutionary processes. We also talked about various concepts of species, and we briefly talked about what do we mean by adaptive radiations. Now we will continue with that discussion, and in today's lecture we will focus more upon speciation. I am Dr. Sudhir Verma, working as Assistant Professor in Department of Zoology, Deen Dayal Upadhyay College, University of Delhi. So in today's lecture we are going to learn about the patterns of evolutionary change. Evolution happens. We have seen so far what do we mean by evolution, what are the sources of variation in the evolution, how evolution takes place, and so on. But now we will briefly see what are different patterns of evolution. In short, there are two patterns which we name as Anagenesis and the Clydogenesis, which we'll try to understand in the coming slides. Further, we will continue our discussion on the topic speciation. Speciation is simply how species form. We will discuss various modes of the formation of species, that is speciation, which are categorized in 4 different categories, that is Allopatric, sympatric, Parapatric and Parapatric speciation. We will try to understand the difference between all these types. Next, we will talk about various barriers or the isolating mechanisms which keep two species as separate. That means the mechanisms which help the species to maintain their identity or integrity. Or in other words, these are the barriers or the mechanisms which help in the splitting of a population in 2 different species, or in other words, it helps in the formation of new species. We'll also discuss about the tempo of speciation, that means the speed, the rate of formation of new species. So let us continue our discussion on these subheadings within this topic. And let us first start about the patterns of evolutionary change. There are 2 basic patterns of evolutionary change which are named as Anagenesis and the Clydogenesis. The word Anagenesis comes from a Greek word where ana refers to new and genos refers to race. The Anagenesis is basically accumulation of heritable changes that gradually transforms a species to another one. It simply means that one species keeps on adapting itself to the environment. It keeps on accumulating various changes, and these changes are heritable in nature. So slowly it keeps on accumulating within the organisms, and ultimately it leads to a new species through transformations. For example, you can see here, if this is a bird which is the original species, it tries to adapt in a particular environment, and during this process it accumulates various changes which are heritable, and ultimately it results into a new species which looks like this. So this has changed to a different species, this. In contrast, there is a phenomena which is called Clydogenesis. The word Clydogenesis comes from a Greek word where kleidos means the branches. Here the splitting of gene pool takes place. The existing gene pool gets splitted into 2 or more separate pools, and these 2 separate gene pools give rise to new species. In contrast to anagenesis, which we saw here, focus on this part of the diagram now. Here, this particular bird splits, the species splits into two different lineages, or in other words, the gene pool of the population here splits into two different gene pools, and these two gene pools give rise to two different species. So here you can see in Clydogenesis there is an increase in the biodiversity, whereas in case of Anagenesis it is the transformation of one species to another one, but it does not increase the biological diversity. So we can say that Clydogenesis promotes biological diversity by increasing the number of species. So all the evolutions, whenever we talk about in terms of new species formation, we are focusing more on Clydogenesis rather than Anagenesis. Here you can understand it in more details. As we just mentioned, Anagenesis is an evolutionary change of a single lineage in which one taxon replaces by another without branching. So this part you can focus upon. If this is an ancestral species named arbitrarily as A, it gives rise to species A in the present day. So there is no branching, but a single lineage is getting changed. So one taxon is being replaced by another one and so on, and ultimately a new species gets formed. Whereas Clydogenesis is an evolutionary change of a particular species in which new species are branched off from a common ancestral species. So here you can see there is an ancestral species which is an ancestral species for species B and species C. This gets splitted off or it gets branched off to give rise to two different species. The anagenesis is also called as phyletic evolution or the progressive evolution, whereas the Clydogenesis is called as the branching evolution. In anagenesis, one gene pool is converted to another gene pool. So from here it's getting converted to another gene pool, whereas in case of Clydogenesis, a single gene pool is splitted into several gene pools. Anagenesis does not promote biological diversity, whereas Clydogenesis promotes the biological diversity since it increases the number of species. Now, having this much understanding about the patterns of evolution, and we know that Clydogenesis is the one which increases the species, let us move ahead on what do we mean by speciation. Speciation is an evolutionary process by which populations evolve to become distinct species. In the words of Theodore Dobzhansky, who coined the term isolating mechanisms, there are two broad categories of speciation. These are called as Allopatric speciation and the sympatric speciation. Now, before we move ahead to these types of speciation, what do we mean by the isolating mechanisms? Isolation or the isolating mechanisms are basically the barriers which do not allow two different species to breed with each other and makes the hybrids. So they help to maintain the identity of a particular species. In other words, these are the barriers or the mechanisms because of which a population gets splitted into two different species which are unable to breed with each other. So, allopatric speciation arises because of geographical isolations. As the name suggests, these isolation mechanisms or the barriers are basically physical or geographical in nature. So, there is an involvement of geographical separation. In contrast, the sympatric speciation involves reproductive isolation. Means there is no involvement of geographical barriers or geographical separation is not there. Instead, they are reproductively isolated. Now, you will understand it more in the coming slides. What do we mean by these kinds of isolation mechanisms or the speciation. Allopatric, the word is made up of two words, that is allo, which means other, and patric, which means the place. A population forms a new species after being geographically isolated from its parent population. An example of such kind of geographical barrier can be a river, that is a water body. It can be a high mountain range, or it can be a large desert, and so on. So let us say if this was the combined population, and because of this barrier, now this population has split it in two different species which are unable to breed with each other. And as the name suggests, they are at two different places. In contrast, in case of sympatric speciation, there is no such geographical barriers. The word sympatric is made up of two words, that is sym and the patric. Sym means the same and patric is the place. So a sympatric speciation is basically a subset or a small group of population which forms a new species without geographical isolation or separation. So in this population, as you can see, there is no physical or visible barriers which is geographical in nature existing in this population. Still, you can see there is two different populations of beetles here. That means the blue ones are going to breed with blue ones only, and the green ones are going to breed with the green ones only. So we are focusing here on the biological species concept, where reproduction is the main criteria to define a particular species. Here, though the geographical barrier is not there, but reproductive barrier still exists. This is the simple demonstration of allopatric and sympatric speciation. With time, let us see if this is the original population and there comes a geographical barrier in between, which splits the species into two parts. And with time, what happens is there is a starting of reproductive isolation, because members of the population which are here, they are breeding among themselves. Members of the population on this side are breeding among themselves, and with time what happens is they do create a reproductive barrier with each other, ultimately giving rise to two different species. Whereas in case of sympatric speciation, if this is the original population, though there is no physical barrier, but within that population there is a small population which breeds among themselves only. So these members are not going to breed with the members onto other sides, and thus it creates a species within the population without any external geographical barrier. Here you can see the characteristic differences between the allopatric and sympatric speciation. As we said, allopatric speciation is basically physical isolation of a biological population by an extrinsic barrier, evolving an intrinsic reproductive isolation later on. Whereas in case of sympatric speciation, it is the evolution of new species from a single ancestral species while living in the same habitat. Allopatric speciation takes place through geographical isolation, whereas sympatric isolation or speciation takes place with geographical isolation or the reproductive isolation. The major differentiation mechanism is natural selection in case of allopatric speciation, whereas the major differentiation mechanism is polyploidy in case of sympatric population, or basically the genetic mechanisms. The speed of the emergence of new species is slow in case of allopatric speciation, whereas speed of emergence of new species is fast with autopolyploidy and slow with allopolyploidy. Allopatric speciation is very much common in nature, whereas sympatric speciation is more common, particularly in case of plants. Now the question comes, how we can identify whether the speciation is allopatric or sympatric. To address this, Cohen and Orr have outlined 4 conditions to be met before speciation could be identified as allopatric or sympatric. The first point is the speciation must have gone to completion. So it should have completed when you are observing that, or when you are trying to identify whether it is allopatric or sympatric. Secondly, the sympatric speciation occurs only between the sister species. It should result in a species pairs with overlapping ranges. And lastly, the knowledge of the past history of the species, especially its biogeographical range, must not allow for past allopatric speciation. So these are the criteria which is being used to identify whether a species is allopatric or sympatric. There are 4 modes of speciation. These are called as allopatric speciation and sympatric speciation, which partly we just saw. What do we mean by allopatric and sympatric? But there are 2 more categories which are called as peripatric and parapatric speciation, which are basically the part of geographical isolation only. So these first three categories, which are named as allopatric, peripatric and parapatric, they are part of geographical isolation, whereas the sympatric one occurs because of reproductive isolation. Let us talk about allopatric speciation, or the geographical isolation, or it is also called as vicariance model. There are 3 modes as I just mentioned, and they differ with respect to degree of geographical isolation, and they are called as allopatric, peripatric and parapatric. Let us see what do we mean by this. We have already discussed this allopatric speciation, which occurs when a population is divided by natural physical barriers such as mountains, rivers, deserts, etc. So there is an original population. The first step is the barrier formation, which is an external physical barrier. And then there is an evolution of reproductive isolation. Ultimately, new distinct species takes place. This is simply the allopatric speciation. But what happens in case of parapatric speciation is, it is a speciation when a population is divided by the budding off of a small, completely isolated founder colony from the larger population. So if this was the original larger population, the first step here is that a small founder population gets separated from this large original population. So there is an entrance towards a new niche by some members, that is the founder members. And then the evolution of reproductive isolation takes place. Ultimately what happens is, even if they come here, they are unable to breed with the original population because they have formed a totally different new species. This is called as parapatric speciation. Moving ahead, there is another category which is called as parapatric speciation. In parapatric speciation, a population at the periphery of a species adapts to different environments but remains contiguous with parents so that gene flow is possible between them. In contrast to peripatric, which we saw in the previous slide, here you can see if this is the original population, the initial step is there is an entrance towards a new niche, just like the previous one, but the barrier is not there. So they have not completely detached from the previous population or the original population. And then there is an evolution of reproductive isolation and the new species do form. But here the difference is that gene flow is still possible with the original population, and that is the main difference between peripatric and parapatric speciation. Now let us just give a few seconds to understand what can be the consequences if there is a secondary contact. That means if there is a barrier which has been removed and the populations are under the secondary contact, how they can respond. So if an original population which saw a geographical isolation and it has given rise to incipient species, then there is a secondary contact. So the first situation can be there is a fusion of incipient populations back into the one species, which is the case where there is no speciation taking place. In case of second case, the incipient species undergo a phenomena called reinforcement. Here what happens is there is a formation of low fitness hybrids, but there is a selection against hybrids, due to which what happens is there are two different species with no hybrids in between. The third case can be there is a stable or transient hybrid zone formation between the two. And the last or the fourth possibility is there is a speciation of hybrids as well. So there is a formation of hybrid individuals and there is a splitting into three different populations or three different species which are having intrinsic barriers among themselves. So these are the possibilities if you bring back together or there is a secondary contact between the two geographically isolated populations. Now let us move ahead towards sympatric speciation, and these sympatric speciation takes place because of the reproductive isolation, unlike the allopatric speciation, which is because of the geographical barriers, and later on they face the reproductive barriers. A sympatric speciation using reproductive isolation can be facilitated by many ways, such as the behavioral, seasonal, temporal, habitat barriers, and so on. So there is a huge list of the barriers which we'll see in a while, which can facilitate the speciation using reproductive isolation or the phenomena called as sympatric speciation. The different classification schemes have been proposed by a number of evolutionary biologists which classify all these reproductive barriers, and there is a different base of classification that has been used by these evolutionary biologists. Four major classification systems which we can refer are the Savage's classification, Myers classification, Mams classification, and lastly the Templeton's classification. Now before we move ahead with all these methods of reproductive isolation, let us quickly have a review about what do these classification systems differ in. The first system that we talked is Savage's classification. This classification method classifies all these reproductive isolation mechanisms or the barriers in two broad categories. One is called as spatial isolation, which are more of external barriers. That means these are the barriers which do occur between the populations, and these can be the ecological one, ethological one, morphological, and the physiological barriers. And the second category is the genetic isolation, which comprises mainly of the internal barriers. That means the barriers between hybrid populations, and these are basically applied on the hybrid, that is hybrid weakness or inviability, hybrid failure to attain sexual maturity, hybrid sterility, and hybrid non-viability or sterility in the later generations. Second classification system classifies according to the geography and level, and it has been given by the Meyer. The first category in that is hybridization, that is maintenance of reproductively isolated hybrid between the two species. Second category is the instantaneous speciation, which occurs through individuals, and they do occur genetically or cytologically. And the third category is the gradual speciation, that means they occur through populations. There they have enlisted sympatric speciation, parapatric speciation, and the allopatric speciation by Parapatric and vicariant speciation. This is another classification system which has been given by Meyer. The third classification system is by Mams. Mams has classified all these barriers according to the stages, and these are basically the sequential stages. He has categorized into two main categories, that is pre-mating or pre-zygotic mechanisms, that means the barriers or isolating mechanisms which prevent the specific cross between two species. The another category is post-mating or post-zygotic conditions. In the pre-mating or pre-zygotic mechanisms, again there are three stages. First is the potential mates do not meet, and the barriers which do not allow the mates to meet can be the geographical barriers, barriers because of distances, climatic isolation barriers, seasonal or temporal barriers, habitat or ecological barriers. Now, if the mates have already met, but they are unable to mate with each other, there might be the existence of the barriers which are ethological or behavioral in nature. Then, even if the copulation has been attempted, that means mating has taken place, but transfer of sperms does not take place. They can be the barriers called mechanical ones or the physiological ones. The second part is the post-mating or post-zygotic mechanisms. Here, the isolating mechanisms which reduce the full success of interspecific crosses takes place. And it also occurs stage-wise. That means the first stage is if the sperms have been transferred but eggs are not fertilized, it is the gametic mortality barrier. If the eggs have been fertilized but zygote inviable, this is called as zygotic mortality. Third case can be the zygote produced F1 hybrids of reduced viability, and this barrier we call as hybrid inviability or inferiority. Next can be the hybrid viable but partially or completely sterile. That means there is a barrier called hybrid sterility. And lastly, there is a reduced viability or fertility in the progenies of hybrids. So F2 generation onwards, there is a issue with the viability or fertility, and this is called as hybrid breakdown as a barrier. So these are the pre-mating or pre-zygotic and post-mating or post-zygotic mechanism classification as given by Mams. The fourth classification is called as Templeton's classification. And this classification classifies the barriers according to population genetic mode, and it categorizes into transilience and the divergence. In transilience, there is a hybrid maintenance, hybrid recombination, chromosomal barriers, and the genetic barriers which do exist between two species. In divergence, there can be a barrier with respect to the habitat, with respect to selection on a cline, that is clinal, and the adaptive divergence barriers. So these are different classification systems for classifying the reproductive barriers primarily. Now we will proceed with the Mams classification. Reason being, it is a bit more structured to understand because all the barriers have been arranged step by step. So it is just like a sequential hurdles. If one hurdle has been crossed, there is another one which has to be crossed. And once two species cross all these hurdles, that means they can breed with each other successfully. So going by Mams classification, let us talk about the barriers in two categories. One is called as pre-zygotic barriers and other one is called as post-zygotic barriers. They are also called as pre-mating and post-mating barriers, but there is a slight difference between these. We will understand this difference when we go ahead with these barriers or the isolating mechanisms in detail. So briefly telling you all, the pre-zygotic barriers do not allow to mate, or if mating occurs, they hinder fertilization. There are three different subheadings within the pre-zygotic barriers that can take place. The first step is the potential mates do not meet at all with each other. So there is a issue with meeting of the potential mates. And if they are not able to meet with each other, obviously there is no further process can take place. Second step is the potential mates meet but they do not mate with each other. Third step is if they have met with each other, if they have mated with each other, copulation has attempted, but there is no transfer of sperms to ovum for fertilization, and that is why the zygote is not being formed, and that is the reason we call these barriers as pre-zygotic barriers. Let us start with the first, that is potential mates do not meet.